Going to be mixing a PCR today, with two primers and two different sets of DNA, and go through the process with some of my co-lab workers, and hopefully we'll get this results by tomorrow. I've worked out my calculations for the PCR. In every well, I'm going to need 10 of the super mix, 0.5 of the forward primer, 0.5 of the reverse primer, and then 8 microliters of water, H2O. And I'm doing two different primers today, so I'll have to mix this twice for both primers. And then multiply by 5 because I'm doing 2 wells for each DNA, so it'll be a total of 4 wells, and I'm adding one on so I'll know that I'll have enough. So let's mix. Okay, I'm beginning to mix the PCR, and I'm turning my pipette to 50 microliters so I can pipette the super mix into both tubes that I'll be placing the two different primers in. The study that we're doing on this is to find a copy number variation on the DNA. Copy number variation is an exact duplicate copy of DNA in the genome of a species that doubled during replication. So it's usually you can have an area on your chromosome that is doubled and not supposed to be there, and it only happens in specific people, but it accounts for 12% of the human genome. So we're looking for this copy number variation so we can compare it to other species. I mean, not other species, but other animals of the same species. I just put in my super mix. The super mix is full of nucleotides, single nucleotides that the PCR will, whenever it heats and cools, it'll go through cycles, and those cycles will cause the DNA to open up, and it'll break the hydrogen bonds between both sides of the DNA. It'll split the DNA down the center, and then when it cools, the DNA, the single nucleotides will line back up on the one-sided plate, and that's kind of the idea of how a PCR works. Putting in my water now. I'm putting in 40 of RNase-free water into each of these. And then I'm going to place in, it'll be a total of 2.5 microliters of primer. 2.5 microliters of primer. So this is my first primer. This is the gene we're studying. We know that there has been, we've actually got a paper published on this particular one, PCCA gene, that it did have a copy number variation in that quadrant. So we're testing it again. So here we go. I've placed in my reverse primer, and now I'm going to place in my forward primer. And when you're mixing this, it doesn't really matter what order you do your super mix in. And that's what we call it, a super mix, when we put all of the fluids in it that we need. But what is important is that you get everything in the right quantity, the right amount. And I always think it's good to pipette the larger fluids that you're going to place in, like your super mix and your water. And when you get those pipetted, it's, you know, it's kind of easier to place your smaller ones in, because you know, you can go down into the fluid and make sure you pipette it all out and kind of mix it when you do that. Now I'm working on my F2 primer, and this basically is testing the same gene, but we know that one gene was duplicated and had a copy number variation in the genome of one of these, and we're testing other samples to see if the copy number is there too, if it's happened to a lot of them in the species of chicken, to find out where the you know, a lot of the copy number variations are happening. Okay, and this is my final primer. Reverse primer is in. So now I have to put it in a plate and spin down. So I'm going to close these up, my little tubes, and I guess I should have put it like that so you could see it real good. But this is basically the fluid of the super mix that will go into the plate. I'm going to centrifuge it down. The item we've got to do here is just mixing it. Okay, just mix the first one. Don't take a lot of mixing for this. And then we bring it over to the centrifuge. Bring it over to the centrifuge. Okay. Gonna open it. This is our lovely centrifuge. Okay. And I'm gonna spin it down. Make sure that this is balanced. Put the lid on, and it doesn't take long to centrifuge it either. Okay. Hit start. Hit stop. Okay, that was simple. Next thing we got to do is get a plate, and I'll do that. These are what the plates normally look like. We don't need this entire plate. I will cut it so we can save some of the plate for later for when we're running these smaller tests. Okay, so just need a small section. Over here, and then cut this right down the middle, because we only need 8 wells to test these 2 primers. And let's see here, I'm going to put it right here, so hopefully you can see it mixing. This is the small little plate that I plan on using, and let's get it close so you can see it. Each well will get 18 of the super mix that I just mixed, and then the other, and then will be 2 microliters of the DNA. That'll make a total of a 20 microliter reaction in the PCR. That's total volume. So here we go. I'm going to grab my pipette and pick up my PCCA. It'll be my first 4 wells that I choose. Okay. Okay, I pipetted in the super mix of my first primer, and now I'm going to pipette in the super mix of my second primer, F2. So the copy number, well, I'm not going to tell you which chromosome this is on, but I will tell you that one of our chicken species, one of our sample, we know for sure, does have a copy number variation. So basically we're testing this one up against that one to see if it also has a copy number variation in its genome too. And that's basically how we do it. If it does have a copy, a CVN, copy number variation, the PCR won't amplify on it very much. So here we go. Now we're going to place in our two different samples, our two different sample chickens. And this was DNA extracted from tissues. And let's see, we're going to go with 6281 first. And yes, okay, 6281 first. That 6281 is just my sample number for my DNA. My sample number for my DNA. I'm kind of mixing. You can see I'm kind of mixing it. Let's see. Where are you? Here we go. I kind of put the pipette in there and I kind of pump it up so I can get the DNA mixed a little bit. And then I kind of mix it when I put it into the well to get it mixed. We also spin down the plate. I have to use a new pipette tip every time because I don't want to contaminate the DNA with the super mix, and then I also don't want to contaminate the super mix, the two different DNAs. So let me put that one back and get the other DNA.
There she is.
Hi, Shannon.
Hey, honey.
I'm doing another PCR.
Did you look at mine?
Yes, everything came out fine on it. But he wanted me to do 2 wells on these, so we're going to do, I'm doing a whole nother one, finishing it up. I'm even recording it. I recorded it because...
How long have you been here?
I've been here since about 11:30.
Oh, your email's so cool.
I know, but Dr. McAdory, I'll explain in a little bit.
I'm talking with Ben.
Okay, hi Ben. But anyway, this is the second DNA that's going in. And once I get this in, I'll cover it with a plastic top and take it downstairs to the computer. And I will place it on the
I'm so sorry, I didn't know you were here.
This
girl in class was like, where are you going now? And I made the mistake of telling her to the library. And she dragged me into the chemistry lab with her. And you know what? And I'm the type that I can't solve problems with another person. I have to be alone and think about it. And she's like talking my ear off. And I'm like, oh, I can't concentrate. And then Oh, is that how you rotate?
Yes. Yes.
So I need to sit there and go to the bathroom.
Yeah. I'll see you in a minute. Okay. Okay. This should be the last well. Okay. 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 Right now we're turning on the PCR machine and we're lifting the hatch back. We're removing the old plate from yesterday. We did a small PCR yesterday that Shannon did and did very well.
Thank you.
Turned out fine. This one is a little bigger. I've got eight wells. I'm doing, and basically on this, if you want to bring it close up, this is these will fit a 96 well plate, and you just have to kind of push this down a little bit, make sure it's okay. Never leave this white thing in on the inside. It'll block your first wells. And if you can avoid using the outer wells, it'd be good, because remember, this is a heat and cool process. The outer wells will have more trouble heating than the inner wells. So if you can at all, avoid those outer wells, it'd be best, when your PCR is cycling. Then we shut it. Then we close the hatch down like that. It's on. I have to come over here. These were the results from your PCR yesterday. You got CT value of 20.7 on one of the DNAs and then a 21.3 on the other DNA. So anyway, I'm going to X out of here. And these are like you won't have a standard curve because we didn't put any standards in it, but after a while I'll show you how to do that. I'm going to X out of here, and if you keep that on the computer, I'll go through the steps with you. Okay, so we're pulling up the iCycler. The iCycler is the button you want to pull up. Okay, yeah, that's it. And then it'll look like that. There you go. Ah. And basically we're doing cyber greener. So we scroll through these until we come upon the one that says...
It starts with an S.
Yeah, cyber greener with an ER though. It's got to have the ER behind it. And then if you look down here, all these numbers, when I click on that, will pop up, and this will show my cycle times, my cycle repeats. This is basically the how we're telling the machine to cycle and what temperatures to go to, set points, and that's what this has. So then we come up here to view plate setup, and then okay, we get a page like this. So then we want to come over here to where it says edit this plate and click on that, edit this plate, and then you get a screen that looks like this. What you want to do is hit erase, this little pencil that says erase, and then erase all the old stuff. So there we go. That's very good for us. So and then we want to hit unknown. And when we do that, we're going to click on our first two unknowns. We did that. I've got two DNAs that are, their number is 6281, and this is PCCA gene. And then we come back over here, we're going to do the second one. And this DNA's number is 6951, and this is PCCA gene. And then come back over here, we get our number 3. This number is again 6281, but this is F1 gene. F1. Okay, and then the next two will be here. And this is going to once again be DNA number 6951. And it is also F1 gene. Okay, so we're good there. Now we have to pick the fluorescence. So we're going to load the fluorescence. We're going to take it off of FAM and we're going to put it onto Cyber Greener because that's the kit we're using, Cyber Greener. And then we take this and click on Cyber Greener here. And then look, they all turned blue because they're all using the same color. So then we're going to name this. We're going to name it after our buddy Raya because this is for her science symposium project on CNV test. Okay, so there we go. There's Raya's name, and she's not here with us. She's in class, I believe, but anyway, so then we're going to hit save this, and it's going to pop up like this. Now I go down to what we named after our doc, and I click on that folder, and I save it in his. So, and then I come over here to quantities. Make sure that I filled everything out right. It looks like I did. Looks like I did. It's showing my 2 wells. Then I come over to edit plate, and I'm going to double check my plate one more time. See if I got all my wells in the right spot where my fluid is. And I do. So anyway, okay, now I think we're ready to go. So what I want to do is hit run with selected protocol, and then I want to change this 50 to a 20 because remember it's a 20 microliter volume in our plate in each well. And then I hit begin run, and then I want to change the name back to Raya's CNV test, and I want to hit user folder. Let's see, Wang 2012, that'll work. Wang 2012, and we're off. We're done. Now we should see some lines popping up over here showing that we've got it. And hi Shannon, this is Shannon.
Hi.
We're in the lab doing a PCR, and there you go. You'll hear that noise come on. When you hear that noise, it's working its way into it, and then you see, whenever I see lines pop up here, I usually go, okay, it's ready. Some of these machines sometimes take about 5 to 10 minutes to heat up. So I know the one we used at Vandy, it was like protocol that you had to turn the machine on for 10 minutes.
There it goes.
And there we go here. We see this, and it'll start making numbers and lines, and this will look like this, and it's ready to go. All right, that's the end of a PCR.
I just wanted to say one more thing.
What?
The most important part of science. Don't forget your pink lip gloss before you do an experiment. It's very important.
Well, you know, I brought mine.
Yes. We both have it.
She's crazy. All right. This film was made by a 4-eyed geek. Thank you.